Hello, everyone, and welcome to Lecture 10, which is a hands-on introduction to GAPIT. We're fortunate to be joined by Professor Alexander Lipka, a professor of biometry in the Department of Crop Sciences at the University of Illinois at Urbana-Champaign in Chicago, and the PI of the Lipka Lab. The Lipka Lab applies cutting-edge statistical approaches to quantitative genetics analysis for crops. This research will result in more accurate quantification of genomic signals underlying phenotypic variation and the prediction of breeding values for agronomically important traits. Prof. Lipka's main research interests involve incorporating functional biology, breeding, and molecular mechanistic knowledge into everyday quantitative genetics and statistical models. So for lecture 10, we'll be covering GAPIT, and basically have statistically optimal approaches for both GWAS and genomic prediction can be conducted for free in the R software package called GAPIT. In this lecture, Prof. Lipka will focus on two widely used R packages for GWAS and genomic prediction. Prof. Lipka will describe the GAPIT R package, which conducts a GWAS and related analysis with minimal user inputs. We will then cover the statistical models using GAPIT and give a demo of a GWAS using the unified mixed linear model. We'll then show how to use the rrBLUP package to conduct a five-fold cross-validation. So thank you so much, Prof. Lipka, and over to you.
Okay, well, thank you for that introduction. And let me just say how happy I am to be here, to be able to give you all this demo and this lecture. Um, as we've done in the previous couple of days, please feel free to chime in if you have questions during this lecture. Um, I love answering questions, especially the ones I don't know the answer to. Um, so what other stuff? So yeah, I'm going to give a demo for GWAS, and I'm going to give a demo for genomic selection. And what I'm going to do is I'm going to do a little bit of motivation for the data set we're going to analyze, and then I'm going to do have like screenshots of the various processes of running a GWAS. And then I'm going to run the demo in real time. And then we're going to go over something similar. I'm just going to go slide by slide what we expect to see with the genomic selection demo. And then I'm going to run it in real time. And I'll also say that thank you, Sumir, for setting up a Google Drive folder. The examples that we go over today are in a folder called In-Class Example, which is actually please run it after the class. And then also for lecture series 9, I had a third bonus lecture that's virtual. And here's the video file for that additional lecture. And please feel free to reach out to me if you have any questions. Um, okay. Yes. Okay, good. And I just looked at the chat and you all have the URL to this drive folder. Great. All right. So, well, without further ado, let's get started. Okay. Introduction to GAPIT and other R packages. Okay. So what we're going to talk about is we're going to run a GWAS and GAPIT. First, I'm going to start with an in-class presentation, then I'll run it in real time. Run genomic prediction using the rrBLUP R package. We'll do it in class, and then we'll run it in real time. So when I was a postdoc, I analyzed maize data, and we were looking at essentially biofortification. So that's, breeding for higher nutritional value. It was a really fun data set to work with, and I loved uh, you know, the benefits for humanity to have increased nutrition. And also the genetic architecture of these traits are much more oligogenic than you would have in a similar trait. So if you run a GWAS on it, you're likely to find a large effect, statistically significant association. But anywho, let me get on with the story. So biofortification, the purpose is to identify target genes with nutritional content. And then you can increase the nutritional value of local crop varieties by selecting on these target genes, and it results in an increased availability of essential nutrients. So The GWAS data that we're going to look at, full disclosure, we already talked about these data on Monday, but we're looking at these compounds called tocochromonols, and they're antioxidants. They consist of tocopherols and tocotrienols. Disclaimer, I'm not a biochemist, so please do not ask technical details about these compounds because I don't know the answer. Um, so the for biofortification, we'd be interested in vitamin E, and alpha tocopherol is the one that we're interested in. Vitamin E is an essential nutrient. In specific population segments, there's suboptimal dietary intake, and deficiency of vitamin E is associated with cardiovascular disease and decreased immune function. So in the data set that we looked at, our maize diversity panel, okay, here's kind of a pie chart showing all the different tocochromonol compounds and what percentage of total tocochromonols is each of these compounds comprised of. And the one in blue, the one with the vitamin E activity, is only about 14%. So what we want to do is, or what my colleagues and my former bosses wanted to do was boost vitamin E levels by increasing alpha tocopherol concentration, by increasing that percentage of that blue pie in that pie chart. Um, so we looked at the Goodman-Buckler diversity panel. It was grown at Purdue University in the United States for 2 field seasons. And then my colleagues at Michigan State, or the colleagues of my former boss, quantified tocochromonols using HPLC. And we fitted a mixed model like I described at the beginning of Monday's lecture to account for field season effects. And then we used BLUPs of our lines. This was before Don't BLUP Twice came out. So if I were to do this again, I'd use blues. But that's a little bit off on a tangent. So we had a total of 20 tocochromonol compounds that we analyzed using GWAS in GAPIT, or analyzed in GAPIT to perform a GWAS. We conducted this on about 294,000 SNPs. And I'll just do quick tangent. One of the questions I was asked yesterday, and I think also Monday, what is the minimum number of SNPs? Um, we determined that for maize, which is, an outcrosser, and you have small LD blocks, we determined that 294,000 was not enough. So it really depends on your species. So this in-class example that I'm going to run in real time, our trait of interest is alpha tocopherol. And I chose that because it has the highest vitamin E activity, and we identified a really strong statistically significant association. Um, um, Sorry, I got distracted by my inbox. Sorry about that. It has the greatest vitamin E activity. So I'm not going to make you watch me run 294,000 SNPs. It would take probably about 30 minutes for everything to run. So instead, I'm just going to run this on a subset of approximately 3,000 markers. And I'm going to use GAPIT. And we're going to use the unified mixed linear model fitted at each SNP. And then we're going to use P3D to save computational time. And if in case you do not remember from Monday's lecture, for FYI purposes, what P3D is doing is it estimates your genetic variance component and your residual variance component prior to running the GWAS. And so instead of fitting a mixed model 3,093 times, okay, instead of running it 3,093 times, it's going to just do all the computationally intensive stuff once. Um, so our data that we have, once you go onto that Google Drive folder and you see everything, you're going to see these files, okay? So this file up here, these are the genotypic data. This is the phenotype data. there's a folder called scripts necessary for GAPIT. So that contains all the scripts that you need to read into R, and then the actual R script for conducting the GWAS. And I've been talking a lot, I've been going all over a lot of slides without asking for questions. So are there any questions? Okay. Okay, just wanted to check. So here's a picture of your RStudio workspace for reading in the GAPIT or for reading in the GWAS file. Um, here's where you download all your libraries. Here's where you read into scripts. Here's where you source in the GAPIT code from online. And then here's where you actually run the GAPIT code. And so once you run all of that, first thing you're going to, you're going to get a folder with a whole bunch of output, right? So we talked a little bit about the QQ plot on Monday, okay? And short version is if the blue dots lie across a diagonal line, it suggests your model's doing a good job of accounting for population structure and relatedness in greater detail. Our x-axis is our observed negative log base 10 p-value for all of those SNPs. Our y-axis is what we expect the negative log base 10 p-values to follow under the null hypothesis. Okay, so we're seeing that most of these dots lie along the diagonal line, providing strong evidence that you know there's no marked trait association for most of the SNPs. And then we see our peak associations as these dots that kind of veer off the diagonal. Here's a Manhattan plot that GAPIT will produce. Our x-axis is physical base pair position along the maze genome, and our y-axis is our negative log base 10 p-value. And over here at the end of chromosome 5, we're seeing a peak association. And we're also seeing something on chromosome 3, which we have not really looked into. And so here's a table of GWAS results, okay? So we saw this on Monday as well. So briefly, we have our SNPs ordered from from most statistically significant, i.e. lowest p-value for testing H naught no marker trait association to the highest. So we see that this marker over here on chromosome 5 has the most statistically significant association. We have an allelic substitution effect of, rounded up to zero point 0.4. Okay. And this is measuring the expected increase in the trait value for each additional non-reference allele. This is so this is estimating the effect of, this is estimating the parameter alpha over here. And then you have your observed trait value, you have your observed genotype of this marker. So any questions before I run this in real time? Oopsie daisy. Oh, I see 3 questions. Hi, is it possible to share the script? Absolutely. The answer is yes. In fact, if you go onto the URL that Sumir shared, if you go into in-class example, everything is there. So if you go to topochronology GWAS, okay. And I'm going to suggest to everybody, please do not run this right now in real time. Please run this on your own. But everything is there. Let's see. Any other questions? Okay, I hear silence. So I'm going to go ahead and run this. Can you all see this font okay? Like, is this big enough? I hear
Yes.
no. Yes? Okay, excellent. So first of all, pretty much the only thing that you would need to change for the GWAS script is you'll need a change your path to your local directory where everything is stored. And that is going to happen on line 10. Um, so let's just run through this. So as you can see, I have nothing in my art workspace right now. So I'm literally running this from scratch. Let me check. Okay. And um, okay, I don't have a significant background, but I'm open to learning. Okay, excellent. Well, we're very happy to have you here. Okay, so okay, so in class example, once you download um, the files from in class example, you're going to get something that looks like this, right? So tocochromanol, GWAS, all right? Um, so first thing I would do is I would change the path, right? So this is the path to this folder, and I would put that into here on line 10. But anyway, let me just get on with it. So here are the prerequisite libraries you need to download. And then here you're setting your working directory. And now we're going to go. So I just set my working directory to this over here, right? So I'm going to tell R to go into this folder and read in all of this source code and then exit. And then I'm going to run, and then I'm going to source in GAPIT 3. Okay, so it's doing its thing. Okay. And note, you will see warning messages. So as the co-developer of GAPIT, let me tell you, don't worry about warning messages, okay? It's error messages you need to worry about. So everything is fine. So here's where we read in our genotype file. Oops, oopsie daisy. Oh no. Okay, I forgot to run this line. Okay, see, even the developer of GAPIT can forget to run lines. So here's how our phenotype data look like. So our rows are each of our individual accessions, okay? All 251 of them. And then we got our sample ID on this column. And then we got our trait value on this column, okay? And then here are the genotype data. View, my G, my dot G. Okay, so we have our genotypic data in HapMap format, okay? And if you're not familiar with HapMap format, please consult the GAPIT user manual. And if you can't find it, let me know. I'll share it with you. Here, briefly, our rows are the different markers, okay? And then your first 11 columns are just kind of like file formats that are stuff that you need for the HapMap format. And then starting on the 12th column, you have your accessions. So, um, you know, for this particular marker, 4226 has CC. I see things on the chat. Okay. Okay. Yeah. All right. Sorry, I was just reading the chat. Okay. So now check this out. Note in here, there is no folder called results. I'm now going to create a directory called results. Oh, and look at that. We got nothing in there. All right. And then I'm going to set my working directory for results. So essentially, I'm going to run GAPIT now. And if all works well, we should see the GAPIT output files being put into results. So let's give this a shot. So I'm going to press the run button. So I'm going to have three principal components. I want to specify that I want to have a 251 by 251 genetic relatedness matrix. I want to run the unified mixed linear model. I'm going to impute missing values of my SNPs with a major allele. Um, That was a choice we made back in like 2012 when we were doing the analysis for Lipka et al. 2013. And this is a crude, rough approximation um, for imputing values, but we rationalized that we were erring on the side of conservativeness. We filtered out all SNPs with MAF less than 0.05, and BIC model selection is what this is doing. So what that is doing is it's doing model selection to determine if you need all principal components in the model for a particular trait. Um, Okay. So let's see. It says that it's done and it gave warning messages, but that's okay. So do you remember how this was empty? Oh, look at this. We now have a whole bunch of results. So, let's see. Um, here's a summary output file of GWAS results. Here's our most significant SNP over here. Um, let's see, our Manhattan plot. Here's our Manhattan plot, okay, which I showed already. And let's see, QQ plots. QQ plot. Oh, here we go. And here's our QQ plot. Okay, and we just created that in real time. All right, does anybody have any questions? I'm looking in the chat. Okay, well, let's now talk about genomic prediction. All right. So let me organize my workspace. We got that there, that here. Okay. All right. I'm going to hide all of my Zoom related things so I can see my slides. Okay. So now genomic selection. So, this is the same data set, okay? So it's the same diversity panel grown at the same locations in 2009, 2010, okay? But this time we're going to look at a different class of compounds with nutritional value, okay? So these are a different trait, a different Y variable in our model, okay? So we're looking at carotenoids, and I'm not sure how much y'all know about carotenoids already, so I'm going to err on the side of telling you stuff you know already. But certain carotenoid compounds have provitamin A content. So carotenoids is definitely another target for biofortification. Um, it's associated with, total carotenoids is associated with degree of orangeness of your kernel color. And that's why I have that picture there. Okay, so now we measured carotenoids for 252 lines of the Goodman-Buckler maze diversity panel. However, maze lines with white kernels do not have measurable carotenoids. So we only analyzed a subset of 201 lines whose kernel color ranges from light yellow to dark orange. And on Monday, somebody asked, what is the minimum sample size for a GWAS or genomic selection? Here, this is the smallest I've ever worked with, 201. And I believe for some particular traits, we even had like 195. We were able to get away with such a small sample size because the genetic architecture of carotenoids is relatively simple. You have a small number of large-ish effect genes controlling these traits. Um, so for other traits, definitely use a larger sample size. Okay, so the data that you have available on that Google Drive for the GS example, you have your R workspace. So instead of, um, you know, waiting like 5 minutes to read in a large file. Instead, a large like text file. Instead, I saved the R workspace from like 10 years ago that contains all of the necessary genotypic and phenotypic data. So this will really save a lot of heavy lifting in the long run. And you read this into R. And then you have the scripts to read in. So you have a folder containing all the scripts with all the functions necessary to be read into R. And then you have the actual R script that's doing the fivefold cross-validation using rrBLUP. Um, so this is how your RStudio space looks like, okay? So here's where you set your working directory, read everything in, and sorry, read everything in. That's really vague, Alex. So here's where you um, read in your libraries. Here's where you load your workspace, that R data file. Here's where you read in the scripts, okay, that has the functions. And then um, I'll go over this in greater detail, but for sake of simplicity, okay, we're going to run genomic selection on only the first 1,000 SNPs, just so that you can have a feel of how everything works. Obviously, in a real data analysis or doing genomic selection, you're going to use your entire SNPs from throughout the genome. And then here's the function I wrote like 11 years ago that does fivefold cross-validation using the rrBLUP package from Endelman 2011. So once you run your fivefold cross-validation, you get this output file, okay? Short version, look at this number over here. It says R average. This is the mean predictive ability across five folds, okay? This is the number that you care about, 0.35. But I will explain all the other things. So this is the predictive ability in each, oh, come on, computer, in each of the five different folds, okay? And then I've never used this before, but I thought back in 2011 it would be useful to know what the sample standard deviation is across these five different predictive abilities. So I included it there. So anywho, take-home message is the predictive ability is 0.35. Across five folds, the mean Pearson correlation between observed trait values and predicted breeding values is 0.35. Okay. Any questions here? Okay, I don't see any questions. Okay, so
Excuse me, please.
oh yes,
I have a question.
yeah.
I don't really have a background in statistics, but I wanted to ask if it's possible that you could give maybe a brief overview of how we got to predictive value.
Okay, so the question is an overview of how it got the predictive value. Yeah. So
Predictive ability, yes.
yeah, of predictability. Um yeah. So what we do is we take our data set and we subdivide it into five equally sized bins. So here we have 201 individuals. So each fifth, in, each of the bins contains approximately 201 divided by five individuals. Okay. And then in four-fifths of our in four out of the five bins, we fit our genomic prediction model, which the Y variable is your trait of interest. And then your X variable is all of your markers. Okay. And then in the last bin, where the data were not used to fit the model, you plug in the marker values into the fitted genomic prediction model, and then you get your genomic estimated breeding value. And then you calculate the Pearson correlation between your genomic estimated breeding value and your trait. And so these five different values we see, these are the Pearson correlations between your observed trait value and the genomic estimating breeding value for each of those five different bins. Does that help?
Yes, please. Is this like you have to repeat this process until you get like a stable number?
Okay, so the question is, do you repeat this until you get a stable number? Um, no, you repeat it for each of the five different bins, for each of the five different subsets.
Okay, thank you very much.
Yeah, no problem. And also I'll say really quickly, if I am happy to share the lectures I gave on Monday and Tuesday, and I I have a slide that goes over this in a detail if some visual aids would be happy with or would be useful for this. I'm happy to share. Any other questions? Okay. So another quality control aspect of genomic prediction is the so-called bias of your predictions. Okay, so um, it is possible to get a genomic selection model that produces, really high Pearson correlation between observed and predicted values, yet the units of measurement of your genomic estimated breeding values is like non-realistic. Like if you're measuring, I don't know, let's say maize, right? Height in maize, it is possible. So a biased model would have, could have a predictive ability of like point 0.8, something really good. But then it's saying that the optimal plant is going to have a height of 300 meters, right? So that would be indicative of a highly biased genomic prediction model. So, what you can do to test this bias, to assess this, is you can make a plot where your y-axis is your observed trait values, and then your x-axis is your genomic estimated breeding values from genomic prediction, and then each dot will be each of the individuals in your data set. And so then what you want to do is you want to fit a simple linear regression model, okay, where observed is the y variable, predicted is the x variable. And then if your intercept estimate is around 0 and your slope estimate is around 1, then you can feel rest assured that your model's not really biased. Like the units of measurement will be realistic. So in this demo where we ran only the first 1,000 SNPs for computational reasons, um, we see that the slope is around zero point 0.9. The slope estimate is around zero point 0.9, and the intercept is around 0.89. So zero point 0.9 is kind of sort of close to 1, but the intercept 0.89, I would argue, is not close to 0. So there could be bias in this model. Um, so any questions here? Oh, I see some stuff in the
Yeah,
chat.
so would you say the bias is coming from the number of markers? If you increase the markers, you have less bias. So, yeah.
Yeah, so yes, Mary. I think a source of the bias here is that we're only using the first 1,000 markers of this data set. Okay, so absolutely, I agree with you. We want to have more markers, and that is likely to reduce the bias. I think also another issue is coverage. So since we're only looking at the first 1,000 markers, we're only looking at like the first chunk of chromosome 1. And so so I think increase the number of markers, but also make sure that there is representative coverage of the entire genome. I think those two factors, I hypothesize, will reduce the bias.
Yeah,
Okay.
so another question, sir. These markers spread across, would you say that they are all accounting for the trait of interest, or how is it defined, the markers that represent the traits exactly?
Right. So let me make sure I got the question. So the question is, how can you make sure that the markers are representative of the trait?
Yes.
Yeah. Um, yeah, this is a good question, and the reason why I am thinking through this answer is um, one of the really beautiful things about genomic selection that makes it so, I guess, for lack of better terms, easy to use, is that we are hoping that our markers are capturing collectively as many possible LD patterns throughout the genome as possible. So what that means is we really don't need to go into GS with a prior knowledge of where the genes are. Um, all what we got to do is just um, see if we are adequately capturing the entire genetic signal of the trait of the genome. Um, and I have a little bit more to say, but does this make sense so far?
Yes, absolutely. Thank you.
Okay. Okay. So the process of determining the adequate number of markers would be to just systematically run fivefold cross-validation, perhaps using this script, right, with an increasing number of markers, and maybe determine the minimum number of markers that gives reasonable predictive abilities, like a predictive ability that is something close to um, what you would see with the maximum number of markers. Yeah. Are there any more questions? Okay. Okay. Well, I will proceed. Um, I will now run this example in R. Okay, great. So let's just navigate through stuff a little bit. So when you download the stuff from the directories, that doesn't make any sense. But yeah, once you like download everything, you'll see this folder called carotenoid genomic selection. Okay. And yesterday, full disclosure, I ran through the script to make sure it was working. So I'm going to delete this folder just to show you that we're honestly redoing everything from scratch. Okay. So I'm going to run this rm list is equal to ls command. And now I got an empty directory. Okay. So here's the script for running genomic selection. Okay. So, oh, come on. Okay. So now I'm going to set my working directory. Going to set this as my home directory. I'm going to read in the rrBLUP package and the mass library. Now I'm going to load my genotype and phenotype data in this R data file, as I indicated on the chat. Oh, I see there's a question. When running a GWAS, is there a specific reason for choosing MLM alone instead of running comparing multiple models? Okay, yeah. So the question, just to repeat, when running a GWAS, is there a specific reason for choosing the MLM alone? I think that the MLM is the most statistically rigorous option out of MLM versus farm CPU versus Blink. So I want statistical rigor in my results, so I'm going to run the MLM. So the other models that I would look into are multi-locus or multi-trait models. So so I do think it is important to run multiple models, but I think that the other models that you compare to the MLM is not another variation of a model where you're testing for the additive effect of a single marker, but to test, more interesting stuff like non-additive effects, maybe, multiple markers, multiple traits. So that's the answer to that question. Um, okay, so. Okay, good. Looks like we successfully loaded all of our genotype and phenotype data. So now I'm going to just read in the scripts with all the functions and go back to my home directory. And then this is how my genotype data look like. So it's pretty much the same data set, same set of markers. And then here's how the phenotype data look like. So again, we have our sample IDs on the first column, and then our trait values on the second column, and we got 201 lines. Okay. So
Sorry, Prof.
yes.
Sorry. When you view your data, you have this thing that pops up like a histogram or a plot that comes up. Like I think it's a histogram. I would like to know how you programmed it in your R to always show you that in the
So the
statistic.
yeah. So this is a great question, Mary. So let's run line 22, which is view my G, and all of a sudden we're seeing these histograms. So Mary is asking what does one need to do to be able to see these histograms? So I'm going to answer this with complete honesty. This is totally brand new to RStudio. When I got my new computer like about a week and a half ago, I had the latest version of RStudio installed, and now RStudio will just give this automatically. So all what I needed to do was just run the view function. All right?
Okay, great. Thank you.
Yeah. Okay, so I'm just gonna do something really quickly. I'm gonna do dimension of my G. So here, in this particular data set, we have 462,703 columns and then 212 rows, right? So this means we got 212 individuals that are genotyped for 462,703 markers, okay? So that is the genome-wide number of markers prior to any filtering, all right? Now, I want this to run quickly in this demo, right? So I'm only going to look at the first 1,000 markers. And the way I'm going to do that is I'm going to, oh, I think I might have said this backwards. There's 462,703 rows and 212 columns, my bad. So, but anyway, I'm only going to look at the first 1,000 markers. So now, after running line 28, we now have only 1,000 rows. Okay, so now the process. Oh, sorry, go ahead. Okay, okay. I don't think that was a question, but if it is, I'm happy to answer it. So the process of randomly partitioning individuals into bins in the process of fivefold cross-validation is a random process, okay? So that means that, a different randomization will result in different subsets of individuals being put into the 5 bins. And therefore, if you run fivefold cross-validation twice on the same data set and you don't control the randomization, you're going to see different results, and that's okay. That's the process of randomization. Okay. So if you want to have control over, making sure your results are repeatable, you can set your seed number through this function over here. All right. Now, to ensure that I get the same results that I've been getting for the past 10 years, I'm going to specify this seed number. So like way back in 2016, I ran line 31, and this is the number that it gave me. So I'm going to set the seed so that the randomization of individuals into our 5 different folds is the same. I see there's a question on the chat. Um, okay, indeed. Um, okay, so now I'm going to create a new directory. I'm going to call it results genome wide. And it was, see, it was just created. And now I'm going to run bifold cross-validation. And it's done. Here are our results. Okay, so let me increase the zoom. And as advertised, I'm getting the exact same predictive abilities as we have in the slides. And that's because I selected the seed number. Okay. And see what else. Um, and then here is the plot of observed values against GEBVs to check the bias. And hey, let's have a little bit of fun, all right? So um, here is what we get if we run all of the SNPs instead of just the first 1,000. So we get a predictive ability. Oh wow, it's even better. Not. We get a predictive ability of 0.46, which is, sorry, I was being silly. So a little bit better. And then bias, let's see. Um, okay. So we're getting, okay, so we're still getting bias in our results. So I wanted the intercept to be close to 0, but it's 0.89, so it's clearly not. And then the slope is 0.94, which is closer to 1 than before. But anywho, I was just looking at that for out of scientific interest. So let me just run through the last slide, and then I think we have time for questions. Um, so we obtained high prediction accuracies, high predictive abilities for this ratio. If a similar model were developed for local African maize germplasm, it would be used to develop maize with optimal, trait values or breeding values for this trait. And genomic selection can be used to expedite the development of local adapted maize germplasm with enhanced carotenoid levels. Okay, so that is my lecture. Are there any questions? Okay.
Sir, and so this, like you're saying that the model can be used for local African maize germplasms,
Yeah.
collections or breeding populations? Which one now? Because yours was the
Right,
diversity panel.
right. So the question is can this be applied to um, diversity panels, or can it be applied to local germplasm? I think that you can take the local germplasm that's being grown in a particular region. Let's see, I'm talking about African maize germplasm. So I argue that you can take at maize germplasm grown at a specific region of Africa. One can use their expertise of African maize germplasm to identify an appropriate training set, and then train a genomic selection model in that training set and use it to predict GEBVs of carotenoid values of locally adapted maize germplasm. And that was kind of a long-winded answer, but does that make sense?
Great, sir. Thank you.
You're welcome. Oh, I see there's a Okay. If possible, share the slides. Yeah. Yeah. So yeah, I would. Yeah. I think I have no objections with sharing slides. Okay. Are there any other questions?
Excuse me, please.
Yes.
Okay, I wanted to ask this question earlier, but I didn't really want to drag you back. There was a particular slide where you had mentioned something like a p-value. Okay, that was meant to be the Pearson's value, I think. You were
So
mentioning something about a p-value and how it represented the... No, it was before this slide.
this slide?
No, it was... I think it was like that, the third slide below, that has like data on it. Yeah, after
Yeah.
this slide. Yes, I think it was this one. I wanted to know how... Okay, would p-value mean Pearson's value?
No, for a p-value, I mean a p-value from a hypothesis test.
Okay.
Okay, so yeah, let me,
Is that
yeah,
as in what y is equal to?
okay. So yeah, so let's go over this. So for this
Thank you.
marker, we have a p-value of 1.92 times 10 to the minus 12, right? So that p-value corresponds to the hypothesis test, where the null hypothesis is that there's no association between PZB022A3.1 and our trait alpha tocopherol. And then the alternative hypothesis is that there is an association. Okay. So the test statistic is going to be a t-test based off of this effect and the standard error of this effect. Okay, so this is going to be a t-test under the null hypothesis. It's going to follow a t-distribution with mean square error degrees of freedom. Okay, so our p-value here, this is the traditional definition of a p-value. This is the probability of observing another test statistic that is as or more extreme than what we observed, assuming the null hypothesis is true. So this is telling us that with this corresponding effect size and corresponding t-test statistic, the probability of observing something as or more extreme than what we observed if the null hypothesis were true is 1.92 times 10 to the minus 12. So that is really strong evidence against the null hypothesis, and therefore we reject the null hypothesis. Okay, and then adjusting for multiple testing correction using Benjamin Hochberg, we get this is less than a false discovery rate of 5%. So I really dived into technicalese with that answer, but that's the p-value and that's how we interpret it.
Okay, so the p-value, I saying that the higher the p-value, the more statistically significant the SNP is.
The lower the p-value, the stronger, yeah,
Okay.
the more significant it is, and to be technical, the stronger evidence against the null hypothesis of no marker
Okay.
association.
Okay. So that's the lower the p-value, the stronger evidence against the null hypothesis value.
100% perfect. Yeah, thank you for that question, Alexis.
Thank you for taking the time to answer.
My pleasure. Okay, are there any more questions? Okay.
All right, we'll give it 30 seconds. If anybody can think of any questions or would like to ask anything, please raise your hand or type it in the chat or unmute and ask.
Yeah, I wrote in the chat that if Professor Lipka could kindly put the slides in the Google Drive if it's possible, so that there's no need of we'll just appreciate that. So it's easy to go to download. Thank you.
I think I am happy to share it. As long as long as it's okay with Sumir, I'm happy to share my slides.
Sure, Alex. Just let me know. I'll update the link, or if you could just email me your slides and I can upload them, but as PDF in PDF format. Then I'll
Okay.
upload them in PDF format. Sure.
Okay. So I'll put my slides into PDF format and then I'll email them to Sumir.
Sure. Then that link where the video is and the scripts for the R scripts is where I'll also upload Prof. Lipka's slides. Is that fine?
Work with me.
Okay, great. So maybe if you guys just want to bookmark that folder so you can always come back to it in your browser. Great.
That's the Google Drive folder.
Yeah, the Google Drive folder for
Okay,
where I
thank
put the
you.
link in the chat. So as soon
Okay.
as when I get the slides, I'll upload them so you can check there within a day or two.
Thank you.
Yeah, my pleasure.
All right, great. I think that brings us to the end of this lecture series by Prof. Lipka. And thank you so much, Alex, for your talks. They've been really engaging and really interesting. I know I've learned quite a bit. And so just to say a big round of applause via Zoom. So thank you so much.
Thank you, everybody.
Great. And please join us. We'll be having the next set of lectures around the 11th and 13th of August with Nelson and Vamsi, where we'll talk a little bit about AI and practical applications of GWAS and plant breeding. All right, thank you. Please do look out for the ABI website and for our social media channels because we'll post the links for the registration to those talks around there. So thank you, everyone. Have a good evening. And thank you, Prof. Lipka. Have a great
All right.
evening.
Goodbye, everybody.
Cheers. Bye.
Bye bye.
Thank you. Good night.